I proposed like maybe we can have more initiatives like these kind of journals that are really nice to be close to the methods and all the things. But it's really nice how the methods are displaying this kind of, because it's just a video and you can see the researcher and you can, it's really nice. I don't know if some older people know, but.
Jovi does a journal.
Yes.
Ah, I see. Yeah, I've seen this journal. Yeah, it's a good journal. I like it.
It's really.
Yeah, that sounds actually like a good thing. I was not familiar. Okay, thank you.
Sean?
Another thing that we were talking about is that you need to be restrict about what your data is telling you and do not try to extrapolate this just to make your paper looks prettier. And then if what you have is just, oh, it's not this, you need to say just it's not this, and then it's not this and it's this, you know, like be strict about what your data is telling you.
Yes, thank you. So keep everything like in the right proportion, right? Like don't extrapolate more than what you can actually say about the data.
For me, the main purpose was to shorten the amount of demanded work, because they explored through high throughput techniques a lot of options, and they eliminate just to focus on the ones that are necessary to work on laboratory. So
yes.
For me, it's to save a lot of time.
Yes, I mean, you don't have power, right, to explore everything in a data set, but to choose. Yes, that's a good point.
We discussed that one of the main factors is collaborative science, because there were people who collect the data and there were people that, you know, like analyze the data and also have controls to validate the research, good statistical analysis, like it's not p-values, and good management of the data and understanding the biases and errors while conducting the research. That's the summary of the course, I think so.
Yes.
Good, thank you.
Yes, collaboration is important. Diversity in ideas, information always brings a new perspective to your work and diminishes bias. So, yes, good. All good.
Anyone else? Anything to add? Jay? Oh. More efficient.
We talk about that they use a big data sets, and also they compare with many of them, and they use multidimensional data sets also to do the research.
Yes, so, okay, all of these things are really good, but I think it's really, in a way, easy to spot them in a paper. I mean, because maybe it's also in retrospective, because we know that those stories worked and what did they do right afterwards. But it's actually much harder to implement it in your own research. And you may think that this is all like, yeah, you know, it makes sense, whatever. But it's very common that people don't follow these things. And, you know, that gives rise to what we've seen in this class also, this crisis of reproducibility, right? So this is hopefully more than just an exercise of, you know, whatever makes these papers successful or reproducible. It's more try to get at least a couple of things from here back to your own research. If it is maybe predetermining what you're going to investigate if you're downloading data, so you're not biased by what you see, and then you follow that, and then you think you had this hypothesis since the beginning and you're testing it, because that happens many times. And, you know, just your team, find a good team with, you know, diversity of opinions and formations. And I think all of these things are hopefully going to help make research more reproducible overall. But it is, you know, it's important, even though, you know, to just be conscious of these things when you do your own, if you use data. Also if you don't use data, I guess, but it's more important in data-driven research in your work to just, you know, remember what makes these papers reproducible, basically.
Thank you, Daniela. Anyone else final comment, or we can just go to the next thing? Okay. Next thing then. So I just sent you, so this class, because it's recap, we don't have the usual questions, so we are just taking advantage of your time and ask you for feedback on the class. So you don't have to put your name if you don't want to put your name. It's in the questions. You can also, it looks like you want to say something directly to me. That's also possible. Any way of feedback, Slack, directly. Always that you avoid physical aggression is okay. You can give us any type of feedback. But yes, let me put the name here, the link here. But it is already in Slack. It should be shorter than 5 minutes, so just answer. It can be anonymous, it cannot be anonymous, it's whatever you want, and we will not take anything personal. Maybe we should, but we are not going to take it personal. So, thank you.
It was the first time trying to come up with a course for a diversity of formations. So, any pointers as to what you would have liked to see, or maybe was or was not useful, would be good, guys. Please.
Yeah. Oops. Good point. So this one is just like a very simple 1 to 10 grading, but at the end of the class, this session, we will do a more open thing where you can just say more things if you have more feelings.
You have many things to say, very deep within. You can just let your mind go. So, thank you. We've been seeing in real life. I'm not going to take anything personal. No, it's fine. No, but today we have, well, just a recap of everything we've seen in this course, hoping that, you know, we can remember the main points that we saw. And after that, well, you have time to do your final activity, right? Get together and read your paper. If you haven't, that's okay. You know that we know that you have a lot of things going on, and prepare the presentations. But okay, first the recap. And first, okay, what is it? What is data-driven research? I want people to answer me. Do you remember? Go.
Well, I don't know if I remember, but
It's been a long course,
what we should the concept we came up with, but the concept I have now after the course is pretty much similar to what I think the term describes, because I think it's a good term. It's research, which mostly is based on analyzing data, sometimes but frequently from available data, but research which essentially tries to make sense of amounts of data which are not easily understandable simply from looking at as a human being who doesn't know how to deal with more than 10 numbers.
That's right. And also usually we don't have a very clear predefined question, right? This is important. Sometimes we just want to generate new hypotheses, right? We want to see what patterns, correlations, and what we note there, basically. So, if you remember, this is exactly what Kawi said, seeks to extract interesting signals by analyzing large data sets usually, and large can be at many different scales, right? As we saw in one of the papers, it can be just one lab generating single cell data, and that's already a large amount of data, or you can be compiling from many sources and make an even bigger data set. Also, we have seen that data-driven and hypothesis-driven research are different approaches to filling knowledge gaps, but they are complementary, right? And we discussed in one of the classes, one of the first classes, there were different points of view, right? Some people said, okay, first I can make a hypothesis and then collect large amounts of data, use the same tools that we've seen, statistics and everything, and then confirm or refute my hypothesis, and I think that's right. But I think in the most, let's say, traditional senses where people refer to data-driven research is just find new questions. Well, I mean, have a broad question and define it more when you see the data. Remember, it makes use of different disciplines, data management, data mining, statistics, so it often requires a big team. Well, maybe not so big, but you require, a team of several members that have different and complementary expertises, because we cannot be experts at everything, especially now. Not nowadays where, there's basically all this data being produced very quickly. And finally, if you remember in the first class, we talked about words and what they mean. We discussed that we needed to be careful with definitions because people can mean different things when they use the same word. And if you go and download data from the internet, then at least you have to make sure that those people are, meaning the same thing that you think they're meaning when you download data. Many cases, this actually is it happens that I thought I was working with this kind of data. It says they're like, oh yeah, no, but they meant something else. So if necessary, contact the authors. Just make sure that you know what you're working with. Okay, remember we had a class on technologies and data generation. What do you remember of that? We saw how to generate data. I told you how sequencing worked. Do you remember? This was a long time ago. Do you remember anything? Do you remember anything about what we needed to take into account when generating data? Remember the biases we talked about? And in that class, I told you about my field, which was sequencing, and all the steps and all the biases that could happen at every step. Remember any takeaway from that day? A long time ago? Yes.
Good God, there were many.
There's sorry. There's another microphone here. Do you want me to take it? I can.
Yes, I remember in the RNA-seq data analysis, you need to be careful about how the experiment was performed, because this changed everything, how the samples were processed, and also which algorithms they used to map and align the sequences.
Yes.
And these workflows impact the analysis, the interpretation, and everything else, because a single change, for example, the log fold change, impact
all the
quantity, and the analysis.
Exactly, and we talked about also papers that maybe made a wrong choice there, in the sense of like their conclusions were not then confirmed by other papers. And yes, indeed. So we talked about exactly that, RNA-seq batch effects, which are again very common. We need to control for that. In data generation, all of these biases can happen. If we don't take them into account, we may fail. So basically, this is what we had as a takeaway from that day. We have to be careful and conscious of all the choices that we're making when generating high throughput data. High throughput data meaning large amounts of data very quickly. That's basically what it means. A lot of biology now is high throughput biology. You should try, if at all possible, to assess the accuracy of every analysis step. So it's always good to have, if you can, even in bioinformatics, positive and negative controls. It's like an experiment in the lab. So, for example, if you have, RNA-seq data, it's an example. Make sure, for example, that genes that should be expressed are expressed. That's a positive control, for example. You just have things that will point you that you have done this analysis correctly at every step, if possible. Because then errors can, propagate, and then at the end, where did you make them is very complicated. So each step, try to check it. It will save you a lot of problems later. And again, this is similar to the before, what we saw before, but be sure to know how authors generate the data if you're downloading it. Because at least in sequencing technologies, read length, protocols of single read or parent read, all of these things that are very technical will completely affect the way you do your analysis. And I don't know how it is in other fields, but I'm sure, at least to me, it has happened to me. I learned late in life. I downloaded sequencing data, I'll just use my protocol, and it's wrong. Why do I have 3,000 million mutations? Because it's all badly aligned, and So just be conscious of these things, because you do learn them in life, but you can be very slow like me and make all the possible mistakes and be like, maybe, I should have read this thing more carefully, or, ask the authors. So yes, just be careful of that. Now, do you remember how we discussed methodologies for data analysis? That was our day 3. That was a controversial day.
I remember that in that session we mentioned about the p-value.
Oh yeah, that was our p-value day.
Yes, and we had to be very careful in what is the meaning of the p-value, and when, in order to don't reject the null hypothesis or accept it.
Yes, so in this session we saw a lot about p-values, and I'm just hoping at least that from this session and overall the class, I guess, you get that we may think we know what a p-value is, and maybe you know the definition from the classical definition, but when you're actually looking at data and getting p-values out, it's not that simple, right? Because it depends on so many other things that we're not taking into account with a single test. Like, remember how likely is that your hypothesis was true in the first place. The p-value can be the same, but if that changed, then your post likelihood that that's true changes a lot. And the number is the same, 0.01 or 0.05. So, yes, so if you are confused about the p-value, this is a good thing, because I am still confused about the p-value. And I use it a lot, but every time I calculate, it's like, what is it really telling me again? And I think the takeaway from here is that if you use p-values, that's okay, because they can be informative under certain circumstances, but they should not be your final answer. They should just be part of a process. When you have to gather more evidence from other sources in order to make your point. Do not rely on just like, oh, 0.01, whatever, p-value, yay, it's true. Or I reject this in favor of the alternative hypothesis because you don't know if you stated your alternative hypothesis correctly. Right, so there's a lot of things to take into account there. So we also talked about, before the p-value on that class, about the initial and exploratory data analysis steps, IDA and EDA. And just basically the emphasis on, before doing any statistical analysis, just clean your data. Clean it, and make sure that the distribution of variables looks like you expect, that you don't have any missing data that you don't know of, that basically you know the data that you're working with. Also, yes, based on that, there's a million ways you can do statistical analysis. You can use frequentist, you can use Bayesian, you can choose among many different kinds of tests. So just make sure that what you're using makes sense. And if you're not an expert in statistics, I'm not an expert in statistics, just find statistician friends that are interested in your project and make a team. So yes, so just be careful, because here I think is where most of the problems with published biomedical literature we know of arise. Just bad statistical analysis or underpowered studies being published and concluding things that they do not have the power to conclude. Okay, and yes, p-values. Think about them. Use them, but with caution. Okay, do you remember what we saw in data visualization after? What makes a good graph or a bad graph? Anyone wants to tell me something? You? No? There's one here. Gracias.
Well, if I remember correctly, it depends on that kind of data. So you have to take in consideration things like if the zero is important or not in your data, yes. It means something. The colors you use, we have discussed a lot about using green for high.
Exactly, intuitive colors.
Yes,
right?
Intuitive colors. And well, I think, and never using pie charts. Although I have some places where I would like to use them.
Yeah, the human eye is bad at judging angles and areas. So yes, pie charts are now discouraged, I think.
I remember that the first thing that we saw in this topic is that think about the message that you want to transmit. And if you think about the message, it's kind of easy to see or to consider which plot do you want in order to represent the message that you want to transmit.
That's right, because you just don't plot data. When you make a paper, you want to communicate something with a plot. You're just like, oh, look at my data. It's like, no, I want to tell you how I saw this correlation, for example. And then you have to somehow communicate that with your plot, right? So it has an objective. It's not just like plotting and choosing the best kind of plot. It's really make sure that you communicate what you want to say. And yes, so colors, the type of plot, exactly as Thales said, that's important. Because, bars, for example, when there's no zero is completely misleading, for example. Pie charts as well these days. And remember, simplicity is preferred to complicated graphs. Sometimes you may have to do 2 graphs for 1 graph. And but I can't plot everything in 1 graph. I mean, you can, but maybe no one will understand anything from you. Or be like, 3 hours looking at, well, what exactly does it mean? So you don't want that. You prefer, for example, in those cases, 2 plots that immediately tell you something. Someone else wants to say something about data visualization? No, we move.
Oh, yeah, I gave up saying what I was going to say because then the slide said, because the point I was going to bring up was related to simplicity. So sometimes as we saw, there are examples of graphs which are overcrowded with information and seem exuberantly sophisticated, but in the end are just hiding simple information which should be presented otherwise in not so fancy a manner. So.
Exactly, and now I think there's a tendency, for example, sometimes newspapers like the New York Times, sometimes they make very nice visualizations, but sometimes like, this is too complicated, and I think they just fancy it, like, look, and then I do a test, and then it moves, and because they're interactive, like, I mean, you don't need, okay, but you don't need it sometimes, so, it's just, I think still simplicity is preferred. Exactly. Just because you can do it doesn't mean you should. Exactly as Mariana says. Perfect. Thanks, guys. Do you remember when we talked about interpreting data? So we have everything. We have our tests, our data. We've, spotted some correlations, did some statistical tests, and now we have to interpret data. Do you remember what we discussed that day? We talked about papers that were, that did some failings in data interpretation, that did not consider maybe alternative explanations for what they were seeing. Anyone remembers? Anyone from that? From last week? I know it's a long time ago.
The arsenic one is absurd. I think it was in this class indeed. Well, you claim for a cell to be able to use arsenic instead of phosphorus, and you don't even resolve the structure of one single molecule to show it has arsenic in place of phosphorus is absurd, in my opinion. So.
And that is the thing, right? If you find something that goes completely against, it completely against like the laws of life that we know, or the principles that we know, maybe you should check twice. 3 times, find other evidence before making these massive claims. And exactly that, we saw some examples of that, but sometimes it's much subtle, of course. We don't really contradict. That's extreme examples, but we should always try to disprove our own assumptions instead of proving them, right? Erika?
I don't know if the narrative potential it's in this one and the data interpretation.
Yes, it was on this one. That's right. So that's right. So it's important in biomedical science, guys, especially if you work with data, massive data, that you try not to get carried away by things you find in Google. That, I think, feeds on the narrative potential. I tell you again, I've already told you, but yes, I work on cancer, and you can't find any gene related to cancer, So if you find anything, you're like, cancer. Oh yeah, of course, it's involved in cancer. So, I mean, there are theories that say that the omnigenic theory of disease is an actual theory that some person put forward, saying all genes are involved in all diseases. Maybe, but is it important? Is my finding significant enough, as you say? Like, do I care, basically? Like, it depends on what you want to do with it. But for example, sometimes, and that goes again with the p-value stuff that we were seeing, sometimes people are like, this is significant, but do not tell you the effect size of the change, right? So the effect size may be so small that you can't really do anything with that information. It's like, oh, okay, what does this tell me? Exactly, so be careful with finding things, for example, in Google now again, that is so accessible and so easy to find information supporting your hypothesis. It's so easy, like anyone can publish anything these days. And if you only find those papers that support what you saw and ignore the others, then there's a risk that you can get carried away. And it's hard. Only yesterday I had a student send me a new plot of data that was very exciting. And immediately, put gene name and anything. You can find, oh yeah, this gene promotes cancer. No, this gene stops cancer. No, this gene is like, I'm immediately completely lost. And if I already had an idea of what this gene did, I make a story immediately, right? So just be careful that it's more prevalent these days with Google and very easily searchable information. So exactly that. Also, that the errors in this data interpretation can arise from many different steps that we saw before. And yes, be careful of the conclusions of your study, especially if they do not agree with the large body of information that we have so far. That should be, a red flag. And if you have extraordinary claims, get extraordinary evidences. Carl Sagan well said, I think. Okay. Now this week we started seeing this. One day we discussed the influence of the scientific community and how we interpret data and how we form our research agendas. Let's say, so do you remember anything from, this was this week, guys. Come on. You should remember from this week. It's not too long ago.
This class we're talking about the things that can interfere in the probability of your paper get published that are not related directly to your work. So like the reviewer preference, the university you are coming from, your positive hypothesis, your positive results,
yes,
and all this type of thing.
Exactly. So all these biases that are, as you say, nothing to do with the actual research, and also the role of people like funders. They give you money, and then, well, you kind of do what they're interested in because they gave you the money. Oh, we also saw, remember, we discussed a paper by Andrew Wakefield and the birth of the anti-vaccine, the legitimization really of the anti-vaccine movement, and what secret interest he had. We saw an example of Coca-Cola paying some scientists to say sugar is not really that bad, come on. So yes, it's important to, sorry, someone wants to say something?
No, yeah, I was just saying that in this class we talked a lot about this part of the importance of the conflict of interest part in the paper, and that generally we just see, oh, there's no conflict of interest in this paper, and then we need to increase this part. We need to really write what is happening to know how to judge this paper.
Exactly. So conflict of interest, yeah, exactly. Now it's mandatory. I think it's been for a long time now to write what the conflict of interest of the authors are, and not only the authors themselves. They also ask you about like your spouse and in the last 5 years and things like that. But sometimes it's hard to see, as we saw the other one, like this was funded by Life Science Institute, something, and that was like Coca-Cola in disguise. So I don't think it's a big problem, at least from my perception. I mean, it exists. I don't think, meaning the things like this where, directly a company, or the tobacco, we didn't see that example, but it happened as well, right? When the company directly funds, like, say this. I don't think it's that obvious, but, with the amount, again, of information that we have now, it's easy to get lost and sway away. So if you read a paper and it's interesting for your research and you're going to take their findings to build upon, your hypothesis, just make sure that you at least try to understand if they had any of these kinds of biases, because that could be really important also for your research, right? Do you remember what these words mean? Reproducibility, robustness, and replicability? Yes? No? You want to refresh? Over there? Atrás? No?
We basically talked about the crisis of reproducibility in science, that in general way a lot of papers are not replicable, and these kind of questions the authority of what is being attested from the paper. And you showed us too that it started a lot of movements to make specific areas more replicable in some sense.
And actually started this, which is fairly new, I must say, of these publish your protocols along with your paper. You have to, it was recent that at least the Nature Publishing Group added this statistical report summary something, and you have to go question by question, like, did you calculate the power of your study? Did you calculate how many animals to use? How did you do that? And how, so, I mean, it's a newish effort by these publishers at least to try and control that. So yes, so these problems were noticed more acutely with the birth of high throughput science, and people started studying, I think, more intensely these words and these different concepts to define whether a paper was or a method reproducible, robust, or replicable. So reproducibility means, if you remember, being able to get the same result analyzing your same data with your same methods, right? So if you go and tell your friend, okay, I used this program on this data, do you find the same? That's replicable. Reproducible. Robust means you have the same data, but you have reasonably different methods, right? So for example, in my field, cancer genomics, when you call variants, you find new mutations, there's different ways of finding mutations, right? There's also different ways of aligning data, as we were seeing, you know. So if you change those things, if your result is true, and if both methods have been, you know, been reasonably done, you should find more or less the same thing. It's never the same thing, but if you know, this mutation is important, you should find it by different methods on the same data. That's what it means. And replicable means things like using a different data set and different methods, can you find the same thing that you claim you did? And I was putting the example of, you know, some people say in breast cancer there are half of the tumors are mutated this way. And if I go and analyze a different data set of breast, do I find the same thing? That is replicable, right? And of course, science builds upon the knowledge that previous people have published, right? We all take other people's results and build upon them. So we need science to be reproducible. And these days, again, there are several things that are impacting that way. And we talked about incentives, we talked about external pressure, how statistical manipulations can also result on badly designed experiments and bad results. And of course, the lack of transparency with reporting data, which again, we said is not really, in many cases, the fault of the scientist. You think you're reporting at least everything important, but when someone else wants to actually come and say, I want to do this, there's not enough information. So with this, again, now we are asked to publish the actual protocols that the people followed in the lab, which I think is a good start. And okay, so here we, I guess, finish with what we've discussed in this course. So undertaking a data-driven research project is hard. It's complicated because it's so full of biases that sometimes we think that finding the truth is really complicated. And it can be, but remember we also saw some success stories where we could not have found those answers without the amount of data that we needed to, you know, collect and find these new vulnerabilities in cancer and these new targets, drug targets. So we need data, we need these projects, and they do teach us something new, and they do advance science as a whole, right? But we need to make use of a lot of expertises and a lot of caution when we undertake a project like this. And of course, yeah, we did see some successful stories to, you know, not give you such a sad impression of the state of things, right? João came and said, give me hope. It's really interesting. When you actually get into this, if you are actually interested in using data, big scale data, it's really, I don't know how to say, you get really into it, right? It's really immersive. I really like it, but I've been wrong so many times in my life that one learns these things. It's like, you know, at least be aware of them. But it's good. It's a good career. If you got a bad impression, please don't. We just need to be aware. Okay, so what next? After we do data-driven research, we had this slide at one of the lectures, if you remember. We had data-driven research. How was it different from hypothesis-driven research? And basically, what I want to make and emphasize here again is you usually take this step and go to a hypothesis-driven approach later. So do not think that when you do an exploratory analysis, you have finished. If you have pre-reported what you were going to do, then you know whether you confirm your hypothesis at the beginning or you actually saw something new, in which case you have to come and do this step again. Right? Okay, so we're going to stop here again with this slide where there's, you know, a lot of fields that use loads of data to move forward. And now we have another questionnaire.
Okay, so this is just an activity, again, for you to reflect. There is a board associated to it. That's the link, but it's here, and it's also in Slack. It was supposed to be by Teams, but it's only 5 minutes, and I'm thinking, don't go to your Teams, just discuss with the people next to you, self-organize. Just don't leave anyone alone. And yeah, put whatever you think is the main points in the board, and 5 minutes working by yourselves and 5 minutes discussing all together. One more minute. let's move to discussion. Who wants to start sharing whatever do you think is the main point here for using data-driven research to get a hypothesis? If I don't see hands, I'm going to start volunteering people.
I think, I guess to think of an example which was not so discussed here, but I think it's interesting how you can use data to try to refute the very presuppositions that you use to analyze data in the first place. I think this is something that is going on very strongly in physics today, because people are really struggling to refute the theories that we have. So it's like people are trying, there are these massive experiments that are gathering massive amounts of data just to find an exception to the theories that we know. And it is one of those things where you kind of work backwards, right? You're like, I'm going to use this kind of theoretical framework to gather data to see if I can find nonsense in some way. I can find something that shows that it is wrong. So I think this is something, it kind of generates hypotheses in this kind of backwards manner, right?
Yeah, that's a really interesting point. I think, I don't know if biology is going to get there because we are so full of exceptions, right? Like every single thing you can think of, in biology, we know an exception, but it's a really cool concept. Anyone else wants to add something? I'm going to volunteer people. And I'm going to volunteer okay.
So we discussed that somehow the large amounts of data we have today, even if they are observations, they aren't observations really necessarily done by the researchers. And that available data may substitute the step in the traditional scientific method of making observations and from observations formulating hypotheses. So to a new alternative path in the scientific method where the step to be done, I think, is to make the available data presented somehow, and by presented I mean it in the broadest sense, so that you can from the presentation you made of it make interpretations and formulate then hypotheses. Differently from when you are doing the observations, the experiments yourself, when you have a more tangible experience of a system which you are studying, so the hypotheses kind of emerge out of the way you're directly interacting with it. And in that case, we already have a system, an amount of observations, but we cannot interact directly with it. So,
Yes, this new way of thinking.
It seems you're off.
This on, okay. Yes, sorry. That has been, yes, exactly, much more popular now in the last, 10, 15 years maybe with this. So yes, so finding the ways to reconcile them both is something that we have to think about very clearly, guys, because you will find both in your research.
I actually think this is a really nice, it's a beautiful way to see it, right? We always go from observations to get a hypothesis. It's just that with big data, that part of the observation, you're just lost in the sea of things. So data-driven research is in a way just trying to present that data in a way that makes sense. So you can do the same thing we have always done. Yeah, I think it's a beautiful way to summarize this.
Everything.
Okay, so Daniela is gonna show you another thing, and then we will have another activity.
Just a couple of slides.
Yeah.
Okay, so another thing, so we've now discussed everything, right, in at least in this round of discussion, how to use data-driven research to do our own research, and how does it connect to hypothesis-driven research. and in the pursuit of truth. But another important point is actually how to conscientiously avoid pitfalls and exercise good practices. So to summarize, I guess, just 3 points of everything we've talked about in this course is, again, understand how your data was generated. And at least, data are very complex, right? So sometimes you say, well, I don't understand everything about my data set, everything that, my colleague gave it to me, and I do not know everything. But at least to the extent of the relationships and correlations that you are interested in. So for example, are those completely confounded by, again, batch effects or something? Just understand how the experiment was done. If it's too big and complex, at least understand it to, the parts of the data that you're interested in. And again, data are very complex these days, so expertise from different fields is important. And again, if you are considering going into this field, either finding a very good statistics support team or just actually studying statistics as a whole degree or expertise, it's a good idea, I think. I mean, if you can do it, or if you can find a very good team that supports you, because a lot of it relies on the tools that you use from there. Again, if possible, I know sometimes this is not possible due to financial constraints, for example, or just very complex data that was collected. And in those cases, I guess you just have to make sure that you did your steps correctly and try to check every step. But if you can, make sure that your results are reproducible. And that can be something simple as, upload your code to GitHub and have it there so people can study it and tell you if they have, questions or comments. If you can repeat your analysis in different data sets, if that's possible. If you find something like, oh, I find that this mutation characterizes this type of cancer, can you find another collection of that type of cancer and repeat your analysis? And do you find that again? And yes, ask collaborators if you have. These days, at least biology, large-scale biology is done by big teams. So if you can ask your collaborators for help checking the data, that would be good. Actually, something just happened to me a couple of days ago where I did a whole analysis of a data set, and I thought I had checked everything that I keep telling you guys about. And then and this is a melanoma analysis because that's the area I work in. And then another colleague in another university that has worked with this kind of data for a very, very long time came and said, yeah, but you included an eighth of your samples are not melanoma samples. And it's like, what? I checked everything, and then, something as simple, now I see it's like, how did I miss that? If I, checked for everything else. So if you have a team, and this still happens, this is something that, it's not something that you learn, and you're just like, yeah, I'm not gonna make any mistake. It just happens. So if you learn, for example, to have a good support team, to ask collaborators for help checking, I've done this, do you have time to help me check if that's correct? Sometimes it's a big ask, but if they're part of your team, they're responsible for your results too. Now if teams publish papers, in theory, every author is responsible for the results. So someone should hopefully check you, help you check. Yes? No? And you also, as we say, you can use data to generate new testable hypotheses. And finally, I would say read from your field, because that's also where inspiration will come for you to form your hypothesis. But be aware of biases and everything that we've talked about in the published literature. And a question mark just to say anything else that you guys remember or have questions about avoiding pitfalls? Okay. Yeah, that's fine. I'm there. Avoid pitfalls and exercise good practices. Any other comments here? Is that everything we said?
We will have, so maybe do the activity, the board, unless it's a question. But otherwise, this is the activity again with the board, and then we can, they can do the comments with the activity. Or is a question?
Yes, he has.
Okay.
No, it's not a question. You asked for other things. I think statistics, you should be careful with the statistics. I think this was one of the most important points of this class,
yes.
maybe. Because we usually...
Sorry, that's probably my fault. Forget to go.
We usually use only the p-value, and this is not enough and everything. So I think this is one of the big messages of this course, maybe. Be
Be
careful with the
confused about
statistics.
p-values. Yes, it's good to be confused about p-values, exactly. They do not mean what we think they mean. At least you know that they're not as simple as people try to be, oh, small p-value, therefore truth. Well, it's not as simple as that. Thank you. Okay, so we have our final activity before the other activities that you already have, your paper.
Yes, so in the same link, the second board is about avoid pitfalls. Same activity, 5 minutes to discuss within yourselves, and then 5 minutes to share your comments with the rest of the group. Like we did 10 minutes ago, like I self-organize. Always that you have someone to discuss. I don't care for this particular one. Thank you. One more minute. Okay, so thank you for the comments here. They are really good. I think this is basically a really good summary of the class. So I'm sharing this as a PDF with all of you if you want to remind it. But I'm glad that you're reflecting about these things, and I think you are having a good takeout from the class. So any additional comments? Do you want to discuss anything in specific? Otherwise, this time I'm not gonna, how you say this, volunteer anyone. We can just move on. So anyone wants to discuss something about this? We are all in agreement that these are good points, and this is basically the takeaway from the class. Yeah, I like that too.
Going in the open science direction, I tend to think that the only good, really good method to avoid it is collaboration and having people outer visions towards your work, because you may, as we suggested here, try to be very meticulous about every step you took and to avoid blind spots but then you get so into your work and you will have blind spots inevitably, and I think the only good way to avoid the pitfalls is to give up on the whole competition idea and start working collaboratively as scientists, because so
Thank you. So sometimes we find people that are with us for one project, for example, and because we work in a research group and that's the person that's there and we collaborate. But if you go down this path of being a scientist as a job, overall, long term, you have to build a team of collaborators that are good collaborators that have the same interests and values that you do, and that takes time. It's not easy to be like, oh yeah, you, my statistician, and you. It's complicated. I at some point, sometimes people try to do this. At least to me, I can define as a bioinformatician, I guess. And people sometimes are like, analyze my data, and it's like, oh, okay, what? And that doesn't work. Sometimes it's an artificial collaboration. But overall, if you follow down this path and you establish what your research line is going to be, then you will meet people. My closest collaborators are not even in Mexico, right? They're all over the world, and that's the beauty of it. But you build this team, and it takes time. But when you find the team that you can rely on, then that's the way that we can move forward. Because it's difficult. This goes to some questions that some of you asked me about. But how do you find a statistician? It's like, well, that's actually not easy. It's true. I mean, it's high demand. The statisticians have, they can work anywhere if they want. So finding someone that's interested in the kind of science you do, sometimes it takes time. But also, that's why you can use these now online ways of collaborating and contacting people, which is easier, along with everything bad that comes with like social media. There's also a good aspect to it, and that's making contacts and keeping contacts with people that are interested in what you do all over the world. So yes, that's something maybe long term, but it's I think the way to do this kind of science as a career, I would say. Okay, that's okay. So anyone else? These are very good points, guys. I really like that. Statistics, lots of statistics, true. Good, lots, p-value stuff, yes, good that you think about that because everyone uses it in data-driven research. And yes, working teams, I guess, that's all. We can share that with you later. Oh yes, sorry.
Just wanted to add something that I was telling on my group, that we talked a lot about the p-values, but in a lot of other statistical methods and analysis, we do a bunch of very arbitrary choices. So you need to have something to support these arbitrary choices that you do the most that you can. And not always is going to be possible, but try to see, oh, if I change this, what happens? So it's not always that you're going to have something very certain, and some of the choices you have to make, like, you know.
It's arbitrary. No, that's true. Sometimes we don't know all the features of our data, but that's exactly the point, right? We make a choice, the most informed choice we can, and then we need more evidence. Exactly that, right? So it's hard. It's not easy. I mean, we can say things here, but when you're actually doing it, it's like, what do I do? Like, I could use these, or I could use these, or I could use this. In some cases, I use all of them and just hopefully they give the same result. And if they don't, then that's a problem, right? And you have to think harder. It's like, why is it not giving me the same result? Yes?
Yeah, I just wanted to add, but interesting questions are always hard. You want to be in the hard line. So do you want to give the summary?
Okay, so just overall summary of things that you already know. But they say that saying the same thing over and over again, it helps with, remembering everything. So the last slide, again, overall summary of the course. Yes, just be careful, make good teams. Do not trust everything you read. Be careful with statistics, which is where most of the errors come from. And be aware of your biases and that of your collaborators if you can. And finally, well, we hope you have learned something from this course. And we have only about 10 minutes before the break. So these 10 minutes would be if you guys have any questions. And you have, we asked for another set of feedback, different questions that you asked, just 3 questions about activities you would like to see or changes that you suggest to this course. And if you have any general questions before we go to the break, now is the time.
Yes, so whenever you are done with this, that I guess is completely optional. As feedback, you want to give it to us. If you have feelings, more strong feelings, or more, like nuanced comments to do. Okay, you can do it also later. Like we don't, it doesn't have to be right now. And the same I was going to say for the questions. So I don't think we are gonna stay in Slack forever, in this Slack. Well, no, but anyway, I'm probably, getting myself out of your Slack just because it's your Slack, but I will be sure to put my contact. So, and I think the same, I will put Daniela's even if she doesn't agree. So you can always contact us with anything that you think we can help you. Liigh. The G there doesn't help, but I think if you...
My only 2 ways of contact really with the world. Email and Twitter. Yes, good.
She's much better replying at Twitter, so if you want a fast communication, do that. But yes, so if you cannot access the questions and you want to go for your break and you don't have questions for us, you're free. Go get coffee and cake and pound the queso.